Okay, this is chapter 18, uh, part 2, on eukaryotic gene expression. So the first part talked a lot about the lac operon and the trip operon. Those are genes that are working inside bacteria, in particular E. coli bacteria. This is going to focus on eukaryotic gene expression that happens in eukaryotic cells, which are cells that have a true nucleus. Differentiation we're going to learn about is when cells become specialized in early embryonic development. Determination are the events that lead to this differentiation. Each cell contains the entire genome, so every single one of your trillion cells contains all of your genes, but only some genes are expressed or actually being transcribed and translated. That's what expressed means, if it's transcribed and translated into a protein or some kind of product. We need to activate or repress genes in various cells. Remember, again, we talked about this in the last video, but it saves energy if you're not making things not needed. For example, we never use some of our DNA in a particular cell. Your nerves never going to make insulin because they don't need them. We use only if needed. For example, a salamander can regenerate a limb, but those genes are only active when you actually need to do that. And then there are some that we use all the time. We turn on when we want to. You can remove cells of a plant to make a new plant. Remember, mitosis clones cells all the time. So there's some genes that are always being active. So in an early embryo, these cells are actually going to talk to each other. They communicate, and that's how they differentiate. Remember when we talked about in chapter 11, cell communication, the signal transduction pathway. There is a signaling molecule that is received by a cell, and it sends the cell, and then it makes some kind of a product. It was called signal transduction, and then response. One cell sends a signal, the signal is transduced, and then ultimately a response is set up. And I'm sorry that that's so sloppy, you probably can't read it. So here's a signal. It's given to the chromatin, which is your DNA. The genes become available for transcription. They are transcribed. Remember, we have RNA processing where the exons are spliced together and the introns are taken out, and it gets a cap and a tail. The mRNA is made into mature mRNA. It's transported out of the cytoplasm, and then it is translated. No longer need the mRNA. It's translated into a protein. Actually, first it's transcribed into a polypeptide, which is just a string of amino acids. Remember that primary protein or primary level of protein production. Then it's folded into an active protein, and then it's sent to do its cellular function. This is kind of a review of chapters 15 and 16. We need to talk a little bit about how eukaryotes regulate their gene expression. And one of the ways they do it has to do with the way they set up their chromosomes. So I want to go back and kind of remind you about how DNA is packaged. Remember, one cell's DNA is about 3 meters long. It's about the size of a car. But we're trying to get into tiny, tiny little cells. And so our DNA is packaged into these histones, and it's wrapped around and wrapped around and wrapped around and coiled and coiled and coiled and coiled and coiled and coiled until ultimately you're into 46 of these tiny chromosomes that fit into the cell. So one of the things that can happen that regulates transcription is modifications to the histones. The histones were these proteins right here that DNA is wound around. First, they can have acetylation. That's actually adding acetyl groups to the histones. Or they could be deacetylation, where you're removing those acetyl groups. Scientists think that acetylation and deacetylation determine how the chromosomes are set up and therefore influence transcription, because they have to have the ability to get in there and make that transcript. So here's your histone, and here's some histone tails, and then you've got your DNA wound around it. Acetylation is adding acetyl groups to these histones. And actually, acetylation of the histone promotes loose chromatin, so it's not wound quite as tightly. And that permits transcription because it can get in and to those genes in order to transcribe it. So what are some things that they can do to the histones to modify them? DNA methylation. This is adding methyl groups. Scientists have found that more methylated the DNA, more methyl groups that it has, the less active it is. For example, the inactive X chromosome that we talked about in genetics in females, remember we have two X's, so a lot of times in order to not get double of a lot of genes, one X is inactive, is really heavily methylated. Experiments have found that in some species, deficient in methylation has actually led to abnormal embryo development. It seems that once methylated, these regions continue to be so through additional cell divisions. Cells keep a chemical record of this. This accounts for genomic imprinting in mammals. There is something we talk about really cool later on in this unit, but it basically talks about how you're methylating your DNA with the things you eat and the things you do every day and all the chemicals you expose yourself to. And that methylation can actually be passed on to your offspring when you have children. And it can really impact their lives. So what you do, when you think about how the food you eat isn't really going to impact your children, but it actually can. We're gonna talk about that sometime in this unit. Here's the methyl group we were talking about here. So other controls on eukaryotic gene expression during the transcription process. Promoters, we think these work like the prokaryotes. Think about the lac operin, but many more of them where the RNA polymerase binds. So there are some ways that the promoters can be affected. Activators and enhancers. Binding site of DNA for the activators turns on non-adjacent genes when the activator binds to an enhancer, which causes the DNA to fold and become adjacent to other DNA. So there's some things that happen during transcription. And this is an example of this. So here's your gene. Here's your enhancer activator that they talked about. Here's your actual gene. And this activator enhancer, they're actually getting closer to the gene because of this DNA bending or folding. And so that actually impacts not only this gene, but it's impacted by this gene. So it's just kind of an idea of non-adjacent genes impacting genes by becoming adjacent to them by that folding. Okay, then there's also things that happen after transcription, so post-transcriptional. Remember that cap and tail we added to the mRNA to protect it from being broken down, the GTP cap and the poly A tail. Also, this is actually a gene expression that we learned about in chapter 15 and 16. Gene splicing, we're actually going to take out the introns and put together the exons. These are the parts that will actually be expressed. Enzymes snip out the introns and put together the exons. So here's an example of this. This is an example of regulating our gene expression because all of these pieces of DNA are never expressed. They're what we call untranslated regions. And then you've got your poly A tail and your GTP cap, which are going to protect this part that is actually going to be transcribed. And then some controls that are translational. Okay, so we've got before transcription, during transcription, now we've got during translation. How long the mRNA lasts. So the mRNA could be degraded early or later, and that depends on the organism and what mRNA it is. Other proteins that bind to inhibit the ribosomes. Sometimes ribosomes are blocked so that the mRNA can't be used. So even though we went through the process of transcription, we can't go through translation because the ribosome is being blocked and the mRNA is useless. And then we have what happens after translation. So eukaryotic gene expression can happen at various stages. It can happen before transcription takes place, during transcription, after transcription, during translation, and even after translation. It just depends on the gene, where it's going to be regulated in there. At this point, we can actually cut the polypeptides into functional units to do its job on their own. Remember, it's not one gene equals one protein, but one gene equals one amino acid, and then those can combine in a secondary or a tertiary structure in order to create a functional protein. Remember, combining those polypeptides is going to actually produce the quaternary structure, and that's going to determine the function. We can save or break down the proteins, determine how long a protein lasts before breakdown. Breakdown enzymes can break it up before it actually gets to do its job. We also have non-coding RNAs. The genome sequence has revealed that protein-coding DNA accounts for only, this is actually, I think, supposed to be 1 to 5% of the human genome. A significant amount of the genome may be transcribed into non-protein coding RNAs. We call these ncRNAs, and many questions about the functions of these remain. We still don't really know. But some of those may actually regulate our gene expression, whether or not we're expressing certain genes. MicroRNAs, miRNAs, complex either degrade the target mRNA or block its translations. It has been estimated the expression of at least half of all human genes may be regulated by miRNAs. This is kind of an important one to remember. Small interfering RNAs, siRNAs, produce similar results as miRNAs. So basically, similar result, different thing. Distinction between the two is the nature of the precursor molecule for each. So they basically have the same function, and these are important ones to remember. And this just kind of shows that these miRNAs or siRNAs, or either way, these can affect whether or not the translation is blocked or the mRNA is degraded. It just happens to do something to it so that it doesn't produce that functional protein. A number of steps in gene expression may be regulated during differentiation. Researchers have used muscle cells grown in a lab to help understand this process. This talks about the MyoD gene. And they've done some work with this to understand this differentiation process and how cells are regulating their gene expression. This is a really cool thing if you get a chance to read it in your textbook. Homeotic genes control other genes that determine developmental sequence. They tell the basic body plan of where everything is going to be laid out. This is an important one to remember. The homeo box is the sequences in the homeotic genes that are very similar in many kinds of organisms. It actually suggests an evolutionary importance because they're so critical to the layout of the organism and the differentiation that happens in the early embryo. So know those two terms. We're going to talk a little about how gene expression influences cancer. Genes that normally regulate cell growth and division during the cell cycle include genes for growth factors, their receptors, and the intercellular molecules of the signaling pathways. Mutations that alter any of these genes can lead to cancer. Oncogenes, we've talked about these before, we're just kind of going back to this. Oncogenes are cancer-causing genes. Proto-oncogenes are normal versions of these that code for proteins that simulate normal cell growth. Proto-oncogenes can become oncogenes through mutations. An oncogene arises from a genetic change or mutation that leads to an increase either in the amount of the proto-oncogene's protein product or an intrinsic activity of each protein molecule. Genetic changes that convert proto-oncogenes into oncogenes fall into three main categories: movement of the DNA within the genome, amplification of the proto-oncogene, or point mutations in a control element or in the proto-oncogene itself. So we've kind of talked about the mutations portion before. Cancer cells are frequently found to contain chromosomes that have broken and rejoined incorrectly. And this just shows the proto-oncogene. Normal growth here. Extra copies of the gene amplification causes excess growth, or point mutations, which is going to cause some kind of hyperactive or degradation resistant protein, so it's not going to degrade when it's supposed to. Oh, man. And these just talk about some chromosomal mutations. This is part of your mutations paper. A deletion will actually remove a chromosomal segment. A duplication duplicates a part, so this here has been duplicated. An inversion actually inverts it, so instead of BCD, you're doing it backwards here, in DCB. And a translocation actually takes a chunk here and moves it to a separate chromosome, and then this one goes to a separate chromosome. All of these can have effects on the offspring. Tumor suppressor genes are genes whose normal products inhibit cell division. Any mutation that decreases the normal activity of a tumor suppressor gene may contribute to the onset of cancer. A mutation that decreases the normal activity of that protein. Basically, you're stimulating growth in the absence of the suppression. Some tumor suppressor genes repair damaged DNA, which prevents a cell from accumulating cancer-causing mutations. So these are the genes you want, the tumor suppressor genes. Here's a couple of known genes. The RAS proto-oncogene results in a protein that stimulates the cell cycle but needs to be triggered by growth factors. Certain mutations trigger even in the absence of the growth factor, leading to an increase in cell division. So this is a bad thing. The p53 tumor suppressor gene protein results in a specific transcription factor that promotes the synthesis of cell cycle inhibiting proteins, activates suicide genes, so apoptosis, programs cell death. Mutations in the RAS occur in about 30% of human cancers, and mutations in p53 occur in more than 50% of human cancers. So those are two identifiable genes that are involved in cancer. And this is just a kind of an idea of how that happens. Cancer can result from the accumulation of mutations, and mutations occur over time. So the longer we live, the more likely we are to get cancer. You know, it's interesting because we wonder if the incidence of cancer is really going up, or if we're just diagnosing it better, or if we're seeing it more because we're living longer. And I think it's kind of a combination of all of those things. I think that there are some environmental things that are leading to cancer more frequently. I also think we're better at detecting it. I think that probably 100 years ago we didn't know what it was, and people died of it all the time. We didn't realize it. But I also think that, you know, over time you're going to accumulate more and more damage, and so the older you get, the more likely you are to develop cancer. And our life expectancy in the last 100 years has gone way up. Mutations contribute to cancer, but we still don't know exactly how a particular cell becomes a cancer cell. This is really important. We don't know this. This is what cancer research is about. We understand that mutations cause cancer, but we don't know how a particular cell becomes a cancer cell. What actually leads to that particular moment where that mutation becomes so big that it's going to become a cancerous and invade other tissues. The development of a malignant tumor is paralleled by gradual accumulation of mutations that convert your proto-oncogenes to oncogenes and knock out those tumor suppressor genes. So here is, it's kind of like a multiple hit thing. So your first hit here in the colon, you've got normal colon, normal things going, and you lose that tumor suppressor gene. You got some kind of mutation there. Now we've got a small benign growth. Now we have hit 2, you have an activation of a RAS oncogene. Now we're like 2 for 2 here. Then we have the loss of the tumor suppressor gene DCC. So now we have 2 losses of 2 tumor suppressor genes, an activation of an oncogene. So now we have a larger benign growth. Okay, remember benign means it's not yet cancerous. Now we've lost another tumor suppressor gene, okay, to more mutations. And then we have additional mutations on top of that, and now all of a sudden we have a malignant tumor. So it's a multiple hit process. The more hits you get, the more likely you are to develop the cancer. That's why detecting the cancer back here is so important, detecting it early, because we might have been able to prevent all of those from happening if we could detect it early enough. An individual that inherits an oncogene or a mutant allele of a tumor suppressor gene is one step closer to accumulating the necessary mutations for cancer to develop. So just having an oncogene does not necessarily mean you will get cancer. Or having a tumor suppressor gene does not necessarily mean you won't get cancer. It just means that if you have these oncogenes, you're one step closer to developing it. Or if you are losing or don't have the tumor suppressor gene, then you're just one step closer to that cancer. BRCA1 and BRCA2 are genes associated with increased susceptibility to breast cancer. Doesn't mean if you have the genes you will develop breast cancer, it just means you're closer to it. Mutations in at least one of these two genes are associated by at least half of inherited breast cancers. Both considered tumor suppressor genes because their wild-type alleles protect against breast cancer and their mutant alleles are recessive. Both protein products function in the cell's DNA damage repair pathway. So in other words, when your DNA is damaged, it's normally repaired, but these have a mutation to that, so it's not being repaired. And that's what's so damaging about it. All that DNA damage is just accumulating even quicker because it's not being repaired. Let's not forget that DNA damage can also be caused by external factors. Smoking, UV light, chemicals, asbestos, things like that. Radiation, even x-rays. That's why they try to limit the number of x-rays you have. And so it makes sense to try to minimize these exposures in our environment. Some known viruses have also been associated with cancers. Epstein-Barr virus that causes mononucleosis is linked to Burkitt's lymphoma. The HPV virus is linked to cervical cancer. It's probably the best known right now because we now have a vaccine for HPV. HTLV1 virus is linked to a type of adult leukemia. I'm not really sure much about that virus, but it is linked apparently. Viral integration may donate an oncogene to a cell or disrupt the tumor suppressor gene, or it may convert a proto-oncogene to an oncogene. So this is answering how on earth does a virus lead to cancer? It could donate. The virus, remember, viruses have their own DNA. And they can actually insert their DNA into your DNA. So they can donate an oncogene. They can actually disrupt the tumor suppressor genes you already have. Or they can take a current proto-oncogene and make some mutations to it, causing it to become an oncogene. And there's the end of our PowerPoint.